Thank you, everyone, for this opportunity. So what I'm pitching is for development of a bispecific antibody against macrophage migration inhibitory factor, or MIF, as well as D-dopochrome T-tautomerase, or DDT, also known as MIF2. So this is our team. I'm a melanoma medical oncologist by trade. I do translational research, and I work closely with Dr. Richard Bukala, who is the preeminent international scholar in MIF biology. So he was also involved in the initial early phase development of imalulimab, which is our first generation oxidized MIF antibody. So why do we care? So the prior attempts at clinical targeting of this pathway have really focused only on half of this pathway, basically targeting anti-MIF or anti-CD74 and ignoring completely anti-DDT potential therapeutics, which have not been targeted to date. So CD74 may seem like an ideal target just because MIF and DDT both signal through it, but in fact, CD74 is found on a lot of immune subsets, particularly T memory subsets, which are critical for tumor responses in memory. CD74 is also correlated with improved outcomes in oncology as well. So theoretically, maybe not the best target. Why we're focused on targeting MIF and DDT is that we know that there is synergism and additive benefits in targeting both to inhibit this pathway. Why a bispecific? It's because we can subvert some of the fallbacks with imalulimab development, meaning we're trying to minimize protein aggregation, and that we're also trying to make this a more streamlined, efficient approach to bring through early phase clinical trials. Additionally, bispecific will allow for more efficient targeting of these cytokines and neutralization within the tumor microenvironment. So MIF in oncology is bad. We know that in terms of autocrine tumor signaling, it increases pathways associated with proliferation, increases tumor-associated angiogenesis, and within the immune cells in the tumor microenvironment, it increases myeloid-derived suppressor cells, tumor-associated macrophages, as well as it increases PD-L1 expression. So in the TCGA data set, we found that high MIF levels are associated with worse outcomes to standard immunotherapies, anti-CTLA-4, as well as anti-PD-1 therapies. What we have shown is that within tumors, ovarian patients as well as melanoma patients, the protein and the mRNA levels of MIF and DDT are highly correlated. Furthermore, in our immune checkpoint treated patients, where we look at CD74, high expression and low MIF expression, so the ratio of these, so low MIF and low DDT relative to CD74 is associated with improved survival in terms of progression-free and overall survival in our patient population. In vivo data using a murine syngeneic tumor model has shown that really anti-PD-1, which is a standard of care for melanoma, only has marginal benefits. However, when we target specific subsets in this MIF-DDT CD74 pathway, anti-CD74 alone in the solid line as well as in combination with PD-1 only improve survival by a fraction. Anti-MIF alone and with PD-1, also same results. Anti-DDT as well as with the combination of PD-1, also again marginal benefit. Interestingly, it's really the combination of anti-MIF and anti-DDT which led to robust overall responses in our animals and tumor shrinkage as well as prolonged tumor memory. This is also synergistic and additive with existing immune checkpoints, suggesting that anti-MIF and anti-DDT targeting is a novel therapeutic checkpoint pathway that can be utilized and is synergistic with existing immune checkpoints. This also represents a very tumor-agnostic approach because many tumor subtypes have high expression of both MIF and DDT. Just to reiterate, we have not made significant progress in this field beyond ipilimumab and nivolumab since 2015. So a lot of the current therapeutics that are being approved are Me-Too monoclonal antibodies targeting the same proteins in this checkpoint cascade. So MIF and DDT combination therapeutic bispecific approach could be that leap forward that we need to make more progress within this field. So what we're asking for is basically funding to create this initial bispecific antibody. We have additional funding already in place to help bring it through preclinical studies. And to make this more realistic, we already have in vivo high affinity CDRs in place against MIF and DDT. Thank you.
More questions? Please.
Can you speak a little bit more about patient selection and, what tumor types and what percent, of those tumor types would have expression of both of these?
So there are multiple tumor types, head and neck cancers, ovarian cancers, hepatocellular cancers. We've also demonstrated the same clinical benefit in mouse models of colorectal cancer as well. And so typically all the immune sensitive cancers we anticipate to respond. It's really a tumor agnostic approach. A lot of cancers have upregulated expression of MIF and DDT.
Great. Thank you very much.